go for is the Florence test. The Florence test used to indicate the presence of colon or not in the given stain. So first of all you have to prepare the Florence reagent. The Florence reagent is nothing, it's just the potassium iodide and iodine in distilled water. So once you have prepared the Florence reagent, then you have to add it into the sample or in the suspect material. So upon the reaction of Florence reagent with the sample, if it contains the semen, it will produce a dark brown color crystal, and upon the microscopic examination you would be able to see that hemine crystal kind of compound in under the microscope, and which is made up of choline iodide. So what happened in this reaction, the iodide part of the Florence reagent used to react with the choline of the sample, of the semen sample, and they will produce a choline iodine that resembles a hemine crystal under microscope, and this is dark brown in color. There is a possibility of getting the negative result that could be obtained when the choline content is very low or stain is decomposed. But this test is very specific to the semen, as it used to give the negative test for the vaginal secretion. So you would not be able to get the positive result. So based upon this test, you would be able to distinguish whether the given stain is the outcome of a vaginal secretion or is it an outcome of the seminal secretion. The next test is for the to know the presence of spermin in the given stain or not. So spermin may also indicate the presence of semen, as it is a part of the semen itself. So for the spermin test, the test that goes for to analyze the spermin is called as Barberio test. So what you have to do is, first of all, again in this you have to prepare the Barberio reagent. Barberio reagent is the saturated aqueous or alcoholic solution of picric acid. So once you prepare the reagent, then you have to add it to the sample, and the yellow coloration will take place, and upon the microscopic analysis you would be able to visualize a needle shaped rhombic, either the needle shape or rhombic yellow color crystals, and these crystals are made up of this spermin picric. So again, based upon this, if you get the positive test, you would be able to say that it could have a chances that the given stain would be semen. The next test that you are going to analyze is to know the presence of prostate-specific antigen in the given stain or not. That is indirectly going to show you the presence of semen or not. So I would like to tell you that apart from prostate-specific antigen, there is one more antigen that is released from the seminal vesicle, which is called as seminal vesical antigen, seminal vesicle specific antigen. So here this is the difference that you can what are their functions. First of all, if you go with the seminal vesicle specific antigen, it used to produce actually two kinds of antigen. One is called as seminogelin 1 and the other one is called as seminogelin 2. They both are responsible to produce a gel-like structure of the semen, and these gel kind of structure are going to hydrolyze later on due to the presence of prostate specific antigen, which is also called as PSA or P30. Okay, so why is it called as P30? Because the molecular weight of this antigen is 30 kilodaltons. So this antigen is responsible for the gel kind of composition of the semen, whereas the reliquefaction within the fraction of a minute used to take place because of the prostate specific antigen that helps in the hydrolysis of seminogelin. And now if you go with its specificity, so this kind of this antigen is also present in other body fluids or in other components of the body like skeletal muscles, kidney and colon, whereas this is prostate specific antigen is usually used to present in urine, serum of males, breast milk and sweat glands. But what happened, these things used to carry very less amount of the PSA, that is not detected under the forensic detection. You can say that it has the low limit of detection in forensic analysis. So thus still it has a chances to give the false positive test because of the presence of these antigen in these fluids, although this antigen is also present in other body components, but these components are not usually taken into consideration while collecting the semen sample. So there are less chances of getting the false positive test in case of this, whereas there are more chances of getting false positive test in case of this prostate specific antigen. So if you go with the screening test, so as they are the antigens, the screening test that can be conducted for the analysis is immunoelectrophoresis. So other tests that could also analyze the presence of these antigens are immunodiffusion, enzyme linked immunosorbent assay, or immunochromatic assay, RNA based assay, but all those have the same principle, that is the antigen and antibody based reaction. So for that you must have that you are required to have the respective antibodies to get the reaction and to know the presence or absence of the given antigen in the given stain. So if we go, the test which you have studied earlier are the presumptive test. To know that actually does the given stain is not is a semen or not, you have to go with the presence of the sperm only. So sperm is the only fluid that is specific to the only component that is specific to the semen. So if you go with the morphology of the sperm, so here it is the microscopic examination, how does it look like and what are the different components that are present in a single sperm. So first of all, sperm is made up of three components: the head, midpiece and tail. And the normal length, the total length of a sperm is around 50 microns, that is from head to tail it is 15 micron long. The head is ovoid in shape and it consists of acrosomes and nucleus. The if you go with the length of the head, it is constitute around 10% of the total length. That is, if the total length is 50 microns, it is around 5 microns. If you go with the thickness, so its thickness is 1.5 microns and its breadth is 3 microns. So length is 5 microns, breadth is 3 microns and its thickness is 1.5. It has a short body and a long filamentous tail. This filamentous tail helps in the mobility of the sperm, and in the midpiece there is a mitochondria, that is the powerhouse that helps to provide the energy for the mobility of the sperm. And if you go with the head, it contains a nucleus, and in the nucleus DNA is enveloped into the nucleus, and the last one is the acrosome. So this the head is capped with the acrosome. This structure is rich in the enzyme to assist in cell wall penetration of egg during fertilization. So here this is the total structure of the sperm. This is a structure is very distinguishably identified under the microscope, but if you have a disconnected head, uh, then it could be confused with the numerous microorganisms like yeast and cellular material of vaginal and other origins. So in you can go for the staining to confirm whether the given disconnected head is actually the semen, actually the sperm, or something some other components. And one more thing, the major importance is in contrast to other cell types, a mature spermatozoa lacks various intracellular organelles such as endoplasmic reticulum, Golgi apparatus, lysosomes and peroxisomes. Now you have to stain the full sperm to know whether the given component disconnected head is actually the sperm or not. So there are a number of staining method that you can go for, like gram staining. The other one is the Christmas tree staining. So here